Hello viewers, welcome to our channel. In this video, we will study the process of glycolysis. The word glycolysis is made up of two words. The word glyco means glucose, and the word lysis means breakdown. The glycolysis is a metabolic pathway that involves the systematic breakdown of glucose to produce energy. The process of glycolysis takes place in the cytosol of the cells. First, we will have an overview of this process, and then we will study the various reactions involved in glycolysis. In glycolysis, one molecule of glucose, which is a 6-carbon compound, is broken down into 2 molecules of pyruvate, which is a 3-carbon compound. And during this conversion process, energy is released in the form of ATP and NADH. The pyruvate formed in this process is further utilized in various energy-producing pathways that produce more energy. The process of glycolysis takes place in 2 phases. In the first phase, the glucose is first converted into 2 molecules of glyceraldehyde 3-phosphate. Since this conversion process involves the addition of phosphate, so the first phase actually uses energy instead of producing it. In the second phase, the glyceraldehyde 3-phosphate is converted into pyruvate, and it is in the second phase that energy is actually produced. Now let's look at the various reactions involved in glycolysis. So glycolysis process begins when a molecule of glucose enters the cytosol. And the first reaction that takes place is the conversion of glucose to glucose 6-phosphate. The enzyme that catalyzes this reaction is called hexokinase. The word hexo means 6, denoting the 6-carbon compound glucose. And the kinases are a group of enzymes that catalyze phosphorylation reactions. So the enzyme hexokinase transfers a molecule of phosphate from ATP to glucose and converts glucose to glucose 6-phosphate. In this reaction, one molecule of ATP is converted into a molecule of ADP. Hence, this step utilizes one molecule of ATP. In the next step, glucose 6-phosphate is converted into its isomer fructose 6-phosphate with the help of the enzyme phosphoglucose isomerase. This step is a simple rearrangement reaction. In the next step, the fructose 6-phosphate is converted to fructose 1,6-bisphosphate, which is the addition of another phosphate group at the first carbon of the fructose ring. This reaction is catalyzed by the enzyme called phosphofructokinase, another kinase enzyme which transfers a group of phosphate from ATP to fructose 6-phosphate. And again in this step, one molecule of ATP is converted into ADP. So another molecule of ATP is utilized in the reaction. Next, the fructose 1,6-bisphosphate splits into 2 molecules, glyceraldehyde 3-phosphate and a dihydroxyacetone phosphate. These both are 3-carbon compounds, and the enzyme that catalyzes this reaction is called aldolase. These all reactions compose the first phase of the glycolysis, and as you can see, energy is actually utilized in this process in the form of 2 molecules of ATP. The 2 compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, are actually isomers and can be interconverted into each other by the enzyme triosphosphate isomerase. But since glyceraldehyde 3-phosphate is further utilized in the process of glycolysis, the equilibrium of this isomerization reaction is always towards glyceraldehyde 3-phosphate. Also keep in mind that every molecule of glucose splits into 2 molecules, so every reaction of the second phase happens twice and produces twice of products. In the next reaction, glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. This involves the addition of an inorganic phosphate group at the first carbon position of the phosphoglycerate. The enzyme that catalyzes this reaction is glyceraldehyde 3-phosphate dehydrogenase, and in this reaction, nicotinamide adenine dinucleotide, which is the coenzyme that is reduced to NADH, which is further utilized in the electron transport chain to produce more energy. Also, this reaction utilizes a molecule of inorganic phosphate. The 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate with the help of enzyme phosphoglycerate kinase. This kinase enzyme transfers the phosphate group at the first carbon position of phosphoglycerate to adenosine triphosphate, ATP. And in this process, a molecule of ATP is generated. In next step, 3-phosphoglycerate is converted to 2-phosphoglycerate. This is a simple rearrangement reaction that occurs with the help of the enzyme called phosphoglycerate mutase. Next, 2-phosphoglycerate is converted to phosphenylpyruvate. The enzyme involved is called enolase, and magnesium ions are also utilized in this reaction. In the final step of the glycolysis, phosphenylpyruvate is converted to pyruvate with the help of enzyme called pyruvate kinase. The phosphate group at phosphenylpyruvate is transferred to ADP and converted to ATP. These all steps compose the second phase of the glycolysis. You can see that energy is produced in the second phase of the glycolysis. Now let's look at net reaction of glycolysis, and then we will understand it in brief. We have one molecule of 6-carbon glucose that splits into 2 molecules of 3-carbon compound pyruvate. There is a net utilization of only 2 ADP, since 4 ADP are used in the second phase, but 2 ADP are also produced in the first phase of glycolysis. So there is a net use of only 2 ADP. 2 molecules of inorganic phosphate are utilized in this step. 2 molecules of nicotinamide adenine dinucleotide are reduced to form 2 molecule of NADH in the conversion process of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This was a brief overview of the process of glycolysis. I hope you find this video helpful. Thank you so much for watching this video. Like and subscribe our channel on YouTube for all the upcoming videos.